Good evening, everyone. My name is Greg Gorga, Executive Director of your Santa Barbara Maritime Museum, and I'd like to welcome you to our lecture tonight, Casting Light into the Deep Sea by Lizzie Duncan. I think I announced this last month, but I'm going to do it again because we're so proud of it. We did receive full 10-year accreditation from the American Alliance of Museums. So it's something we've been working on for about 3.5 years. It shows that the Maritime Museum, the Santa Barbara Maritime Museum, practices best practices in the museum world. There's about 33,000 museums across the United States, and less than 1,100 are fully accredited. So we're very proud of that. The staff, our board of directors, our volunteers worked very hard for that accreditation, and I hope you're proud of us as well. I want to thank our good friend Marie Morris-Rowe. I know you're out there, Marie. Thank you very much for sponsoring our lecture series. Greatly appreciate that. I want to tell you about a few things coming up here at the museum. And first of all, I'm very proud of, let me get this forwarded to the next slide. Very proud that our Maritime on the Move program, which we created about 2 years ago through our education department, has received 3 different recognitions for that program. The American Alliance of Museums awarded it as a finalist as one of the most innovative education programs nationally this past year. We were nominated for that. The Council of American Maritime Museums invited our staff to present on this program at their annual conference. And it's not moving to the next one. And just recently, the California Alliance of Museums gave us their Secretaries for Public Education for Sustainability Award. So really proud of our staff for putting that together for that program. We did just have 300 plus kids go through that program here in Santa Barbara with the Santa Barbara City Parks and Rec Department's nature camps. And in fact, on a bunch of those classes, our good friend Jean-Michel Cousteau participated and told these young students about whales and the Santa Barbara Channel and the islands. So that was really special. Also want to let you know about our current exhibit, Heritage, Craft, and Evolution, Surfboard Design, 1885 through 1959. That is up through October 30th. We have a wonderful Scape art show. These paintings are for sale. Scape is the Southern California artist for protecting the environment. We did this last year. It's on. Unfortunately, we weren't able to do that show here in person because of COVID, but it is online. And so that on for sale right now. There's about 150 pieces of art. We've been doing this Scape show for about 4 years. I got to tell you, this is the best artwork I've ever seen in that show. It's just beautiful, and 40% of all the proceeds from the art sale goes to support the Santa Barbara Maritime Museum, our education programs and exhibits. So please support the museum by getting some beautiful new art. It's all maritime related. A lot of paintings about Point Conception, the harbor, our beautiful coastline here in Santa Barbara. So support the local artists, support the Maritime Museum, and get something new for your walls, which I'm sure you're tired of looking at what's on your walls after a year and a half of COVID, right? We also are doing sea glass pop-up with vendors selling sea glass artwork on our patios on Sundays. We have 2 more of those, the 22nd and 29th. That's from 10 to 5 on our front patio. On Saturday, September 4th, we'll have another 2 artists out there doing a pop-up sales event on our patio. And then September 11th, we're going to do Girls in Ocean Science. This is another program our education committee and staff have come up with. And this one is designed to encourage high school women to pursue marine sciences in college. And so they'll get to go out on the water, work with some local naturalists, and learn about our marine environment and how to take care of it and what careers are possible going forward. They could learn from Lizzie tonight. And then September 16th, our September lecture is the Charles E. Mayer Desalination Plant by Malcolm Hamilton and Galen Fair. And then in October on the 21st, our lecture is Found, a DDT dumping ground near LA and Catalina by David Valentine. A lot of you may have seen this story on the front page of the LA Times about all the DDT that was dumped into our waters years and years ago. Hope you follow us on Facebook. Hope you get our monthly currents and newsletters. You could sign up for our weekly mailings very easily at sbmm.org. That's where you see the Scape Show as well. Very bottom of the homepage, you can just put your name and email address and you'll get all these updates. I want to thank all of you who are members of the Santa Barbara Maritime Museum. We could not do all this without your support. I especially want to thank those of you who are members of our Navigator Circle and our Planned Giving Flagship Society that have us in your Planned Giving. Your support makes our work possible. And we continue to do our happiness hours out on our front patio. We got one more next week for our Navigator Circle members. If you're a regular member of the Maritime Museum, you can purchase tickets for that as well. We have cocktails, food from Uncorked, beautiful view of the harbor. That's from 5 to 7 next Thursday. That'll be the last one for this summer. And then do remember that this is a Zoom webinar. We are being recorded by TV Santa Barbara. Thank you out there for that. This will be up on our website in about 1 to 2 weeks. So Lizzie Duncan is a research ecologist with the National Oceanic and Atmospheric Administration, NOAA, Channel Islands National Marine Sanctuary. She's been part of the sanctuary's research team since 2017 when she first joined as the California State Sea Grant Fellow. In 2018, she first dipped her toe into the deep sea world as a co-coordinator of the West Coast Deep Sea Coral Initiative, a nationally supported and multi-year research program. As she became more acquainted with this mysterious ecosystem through her work, her knowledge and fascination of the deep sea grew alongside her experience in organizing and leading field work in one of the harshest environments on Earth. Now, as a full-time research ecologist for the sanctuary, she continues to coordinate deep sea research, develop and manage related education and outreach projects, and participate in several other sanctuary research studies, such as characterizing sanctuary visitor use and evaluating the connectivity of the sanctuary with other habitats on the West Coast. Lizzie relishes opportunities to conduct field work in waters around the Channel Islands, be it in depths easily accessible by scuba or remotely operated vehicles. She earned her bachelor's degree in marine biology and master's degree in biology from California State University, Long Beach. Her undergraduate and graduate research focused mainly on how climate change may impact the ecology and physiology of marine organisms in rocky intertidal zones and shallow waters of the Great Barrier Reef. Lizzie was awarded a National Science Foundation Graduate Research Fellowship that supported her graduate research on endangered black abalone at Stanford University's Hopkins Marine Station. She also received an East Asia and Pacific Summer Institute Award to broaden her research at James Cook University. I don't know how she did this, she's so young, all of this, where she investigated the effects of sublethal temperatures on epaulet sharks. I hope I pronounced that right. Lizzie's overarching research interest is to understand the role that rising temperatures play in shaping how key species live and survive in already extreme environments. Having explored some of the shallowest, she's excited to tackle questions and challenges in the deepest. Please join me in welcoming Lizzie Duncan.
Thank you, Greg, for that wonderful introduction. And thank you so much for having me. I'm really excited to be here tonight and talk to you about some of my work on the West Coast that relates to deep sea corals and sponges. So I would like to start off tonight by asking you all to close your eyes and imagine what the deep sea looks like. Down at the deepest depths near the seafloor, and if you're thinking it probably looks similar to the inside of your eyelids, you would be correct, but imagine you could bring a light with you and look around. What are you imagining? Is it a bunch of dark mud, maybe some rocks, maybe an underwater volcano? If you open your eyes, maybe it's something similar to these fish with giant teeth, maybe some giant scary squids that have inspired some science fiction movies like 20,000 Leagues Under the Sea, some of which those creatures are actually not too far off from what we're finding today. But I'm going to take a guess and say that many of you maybe did not imagine a vibrant, thriving habitat of many different colors, purples, pinks, reds, of different corals and sponges of different shapes and sizes. So these habitats are made up of invertebrates that live on the seafloor, and these organisms are going to be the focus of my talk. So although those other organisms are definitely worth mentioning here, we will be focusing on some more sessile invertebrates. So backing up again, very little is known about the deep sea. Although many of you may be familiar with the statistic that 75% of the Earth's surface is covered by the ocean, there's actually 95% of the living volume space on Earth is actually the deep sea. So in those three dimensions, it's actually 95% encompasses deep sea ecosystems. The deepest part of the ocean is an infamous seafloor feature, the Marianas Trench. And if you were able to fit Mount Everest at the deepest point in the ocean, there would still be about a mile of seawater in between the peak and the surface of the ocean. So that's pretty darn deep. And we do know a few things about the ocean, though, that the average depth is about 4,000 meters. Only 3 people have been to the deepest part of the ocean in a submersible. And not that it's a competition, but if anyone happened to casually mention that to Jeff Bezos, we might be able to receive more resources and attention in this kind of area. The pressure at these deepest depths can be greater than 1,000 times than what they are at the surface. The temperatures hover just below or just above freezing, so it's quite a frigid environment. And it's very dark. Only 1% of the light that reaches the surface of the ocean actually makes it down to about 150 meters, and 0% makes it below 1,000. And if the average depth of the ocean is 4,000 meters, much of the ocean is very dark and cold. The main source of energy or carbon flow into this ecosystem is actually in the form of marine snow. And this is what that looks like, exactly what you might imagine, a very dark snowstorm. And marine snow is formed in the lightest depths, many layers above the deep sea. And it's composed of plankton and other zooplankton that have decayed and are starting to sink. And they're kind of glomming onto other bits of silts, fecal matter, other dying organisms, and this organic matter is the main source of carbon into the deep sea. And that's important as I talk to you about what are deep sea corals and sponges. So many of you might be familiar with their tropical shallow water counterparts, but deep sea corals and sponges are indeed related to those tropical water corals and sponges. They are part of the phylum Cnidaria. They are made of a bunch of these little polyps here seen in some of these photos I have. And they share a common skeleton, some of which are made of calcium carbonate, which are the hard, stony corals. Some of it are more proteinaceous, and so they're soft sea fans. And they come in a number of different shapes and sizes. The majority of them like hard substrate to attach to and grow. But there are some species, like the 2 on the right side of the screen, that actually can live in soft sediment, and those are called sea pens. About 2/3 of all the diversity of corals are found in the deep sea, and so they're just as diverse, if not more diverse, than the tropical shallow water corals that you can see snorkeling or scuba diving. And so to also give you an idea of some of how they differ from the shallow water corals, you can see on the left there are the shallow water coral reefs in the tropics, and these particular corals have a symbiotic algae within their tissues. And so their main purpose when they build these large reefs is to intercept as much sunlight as possible so they can benefit from that symbiotic relationship. And that's a main source of fuel and energy for tropical deep sea corals, or tropical shallow water corals. So they build these mound-like, boulder-like reefs where they're trying to intercept as much light as possible. But in contrast, deep sea corals and sponges tend to form these forest, grove-like reefs of tree-like branching structures because they lack that symbiotic algae within their tissues, and they rely solely on filter feeding as their sources of energy. So that marine snow plays a really important role in how deep sea corals and sponges grow and survive in the deep sea. It's a similar story with deep sea sponges. They're related to those of the shallow water tropical corals and sponges, And they rely solely on filter feeding, although there is an interesting adaptation in some deep sea sponges where they've actually evolved to be carnivorous. So instead of passively filter feeding, they've evolved little barbs or sticky appendages that a copepod or some other crustacean swimming by, if they were to encounter that, they would get stuck. They secrete digestive enzymes, and then they absorb the nutrients and the carbon that way. So it's a whole different ballgame in the deep sea. The energy input is limited, and so they've really had to evolve interesting techniques to be able to survive. And you'll see in the upper right-hand corner, some of these sponges are stalked, and again, that's to get up off the bottom to increase the current that they're intercepting for filter feeding. And so the deep sea environment is very cold, it's dark, and it's food limited for a lot of these organisms. And so they grow really slowly. Their physiological processes are slowed down because it's so cold, they're food limited, and they can grow on average in an annual basis of between 2 and 20 millimeters per year, so it's pretty darn slow. And on the left, you can see 2 different types of corals. The top is a cross-section of a soft gorgonian, and on the bottom, that's a stony coral known as Lophelia pertusa. And you can see they lay down their layers of their skeletal material over time, so we can kind of count them similar to tree rings, and we can age them in that way. And then on the left, this is a black coral, and the oldest coral dated to present day is over 4,000 years old. And so because the ocean environment, especially down deep, is very stable, they can grow meters and meters tall despite growing so slowly. So this is a black coral. That's not the 4,000-year-old coral, but that is the same type of black coral. And so why do we care about them? And it's because they provide a lot of services that humans benefit from. So the main way is that they provide countless other species habitat in which they need to survive and grow. And it's a lot of species that we care about, and a lot of them we like to eat. From shrimp and crab to flatfish and redfish, deep sea corals and sponges provide them habitat, shelter, food, and a number of other services. And NOAA defines deep sea coral and sponge habitats and ecosystems as essential fish habitat, which means that healthy and thriving habitats like these are necessary for the survival, reproduction, and growth of these species that we like to eat. And similarly, because they attract different organisms and they need them to survive and grow, they create these biodiversity hotspots within the deep sea. And so in an otherwise desolate kind of environment, they are an oasis of life. Also applicable to humans, some species contain metabolites or molecules within their tissues that have great potential as biomedical compounds that we can use and harvest for our benefit. For example, these 2 pretty unassuming sponges. The top yellow sponge has a metabolite within its tissues that has antibacterial activity that can be used to help combat drug-resistant MRSA, or antibiotic-resistant bacteria. And then this green sponge below, it's about the size of a golf ball. This has metabolites within it that can selectively target pancreatic and ovarian cancer cells. And the key word there is selectively, because those are the only cells within the body that it will actually attack and destroy. So there's a lot of potential in the deep sea, and there's a lot of reasons why we should care about these organisms. But of course, anything that's worth valuing is also likely threatened. And there's a whole number of threats to deep sea corals and sponges, many of them which are anthropogenic. And so bottom-tending fishing gear is the most chronic and critical threat to deep sea corals and sponges. Other disturbances such as oil and gas exploration and extraction, marine debris, and of course climate change. In the upper right-hand corner, you can see an image of a net, and that's called a trawl. And so this net gets dragged behind a boat, and the doors open up, and it pretty much drags along the bottom of the seafloor, and that collects everything in its path. So it's definitely good at catching fish, but it also means it's also really good at catching deep sea corals and sponges, for example. So in the photo below it, that is bycaught or bycatch, looks like a prinoid. And from his haircut, this looks like this photo was taken maybe in the 80s. And so bycatch is also a way in which we can learn and study about deep sea corals and sponges, unfortunately, if they're brought up to the surface. And this is an issue because deep sea corals and sponges are incredibly long-lived. So if they're disturbed, They may never recover within our lifetime. It could take decades, it could take hundreds of years, but we don't know a whole lot about how they recover yet. The other threats that include climate change include increasing temperatures as well as acidifying waters. This example on the screen is of a stony coral, Lophelia pertusa, and on the east coast, where the waters relative to the west coast are a little bit more basic and they're more stable. And so Lophelia will grow on top of itself. So as it grows, new skeleton lays down more material and it continues to build a reef. On the west coast, however, because we have a lot of upwelling, it brings in acidic waters from the bottom that are nutrient rich and full of oxygen, but it means that the calcium carbonate within their skeletons tend to get pretty weakened. And so as these polyps grow, they tend to break off and turn into rubble. So that's an example there where a lot of the Lophelia, as they grow, they tend to crumble. So they don't build reefs, but they are still important habitat for now. So if those are the threats, how do we manage and protect them if we know they're important to us and they're beneficial to fish and things that we care about? And the two main ways are through fishery management plans and marine protected areas. So there are many fishery management regions within the US, which is the map on the left. And within a management region, there are fishery management councils, which create and enforce area-based closures as well as gear restriction closures. And so the gears, like bottom trawls, if those are restricted outside of certain areas, that benefits the fish for those fish stocks, but it also benefits deep sea corals and sponges indirectly. There are also national marine sanctuaries, which is the Channel Islands National Marine Sanctuary is where I work. There are 5 on the West Coast, but there's a number of other sanctuaries and monuments within the US. And we protect deep sea corals and sponges a slightly different way through the prohibition of seafloor disturbances. And so we prohibit oil and gas exploration and extraction, cable laying, things like anchoring. So anything that could disturb the seafloor, that's something we prohibit. We do not regulate fishing, so we protect them in a slightly different way. But these are the two main ways in which we offer protection to deep sea corals and sponges. But how do we protect them if we don't know where they are? Like I said, the percent of the ocean floor that we've explored is 5% or less. So the first thing we have to do is some research. So we have to figure out where they are. And the deep sea is a very harsh and remote system, and so we need large ships to be able to withstand some of the ocean conditions, large swell, strong winds. And they need to be able to carry the technology that we need, such as remotely operated vehicles, autonomous underwater vehicles, and so on. And some of these ships are capable of mapping. And mapping the seafloor is like painting the ocean floor with sound. These ships emit acoustic signals, and the signals that they receive back can tell us a few things about the seafloor. How fast it returns tells us something about how deep the ocean is, and how intense the signal returns kind of tells us how hard or soft the bottom of the ocean is. And in the lower right, you can see some bathymetry there of the seafloor that can kind of give us an idea if something's very rugose, if something's flat and likely muddy, and that can give us a clue as to where we might want to look for deep sea corals and sponges. The next step is usually visual surveys. And so with these same vessels, we employ or deploy remotely operated vehicles, autonomous underwater vehicles, towed cameras, a number of different ways to get eyes on the bottom of the ocean. On the lower left, that's an autonomous underwater vehicle. Those are pre-programmed to run tracks, so they're not connected to the ship after it's released. And remotely operated vehicles are tethered to the ship, and those are actually driven by ROV pilots and engineers in real time. Later on, I'll be showing you some video and some footage from highlights from the Ocean Exploration Trust's exploration vessel, the Nautilus, and their 2 ROVs, Argus and Hercules. So this work that I'm going to be talking about is funded by the West Coast Deep Sea Coral Initiative, of which I am a co-coordinator. And you can see on the right, there's a map of all the work that we've done on the West Coast. It's a 4-year research program, and we've gotten together with experts from all across the nation, and specifically on the West Coast, to determine what our biggest data gaps are and what our needs are for conservation and management of deep sea corals and sponges and their related species. And so the work I'm going to be talking to you about today, like I said, we partnered with the Ocean Exploration Trust, and we used their exploration vehicle, the Nautilus, to do some dives within the Channel Island National Marine Sanctuary. This is the sanctuary just off of Santa Barbara and Ventura counties, and we have a few MPAs within the sanctuary as well. And this was in October of 2020. And normally, we get to go on the ship as PIs, research scientists, and we work with a number of engineers, pilots of the ROVs, and communication specialists, and we get to be on the ship, and it's kind of like for comrades. We have 2 shifts. I had the midnight to 4 a.m. shift and the noon to 4 p.m. shift, and so usually when you have those odd hours, you all have those odd hours together. And so you eat together at midnight, if that happens to be when you eat. You wake up at the same time, you can essentially commiserate. But due to the pandemic, for the safety of the crew, we did not board the Nautilus this time. And this is more of what my control room looked like. This is from my office at the Channel Island Sanctuary boardroom, actually. So I had all these nice monitors. I was pretty lucky. And thanks to the telepresence system on the Nautilus, they were able to send us in real time the ROV footage, and we were able to guide the research from our home offices. And although that doesn't seem ideal, it was an extremely successful mission because we were able to do this from home. We were able to include a lot more experts and PIs from all across the West Coast. That would not have been possible otherwise, just due to limitations of space on the ship. And so we had people from the National Marine Fisheries Service, we had people watching from the Bureau of Ocean and Energy Management, all in real time to help us guide this mission and answer questions in real time. So we did several dives within the Channel Island Sanctuary. We gathered video, we gathered images, samples, water samples, and these are all the dives that we completed in about 9 days. And today I'll be highlighting a few of the most exciting discoveries that we made. West San Miguel Shelf, which is on the western side of the sanctuary near the San Miguel Island. There are 2 reefs that are probably the best studied reefs within Southern California with respect to deep sea corals and sponges, Footprint Reef and Piggy Bank. And so we went back there to do some photo mosaics and capture some additional images of places that we are already well studied. And then we went a little bit deeper, which I'll talk to you about too, to kind of extend where we have done the surveys. So first, I'd like to talk to you about San Miguel Shelf. And so on the right, you'll see a map, and this is the bathymetry like I talked about. And this is shaded in a way where you can see the relief. And so one of the reasons we were really interested in San Miguel is because of all of these really interesting seafloor features in the geomorphology of the hard substrate there. So they're kind of swirly, twirly, high relief areas. And we had previously done surveys there with acoustic technology, and we found that there were a lot of fish in the water column, but we couldn't put eyes, we couldn't see them, so we didn't know what kind of fish they were. And in previous surveys, like on the photos on the left, you can see that there, in some areas, there's 100% live cover. So it's another really vibrant place, so we wanted to go there. So this video is being taken by the ROV Hercules, and we're about at 120 meters, and we are on some of those really interesting rugose rocky outcrops that had a lot of corals and sponges growing on them. And this is going to be coming up some Lophelia pertusa, that stony coral I mentioned. And because of those acidic waters on the west coast and how it fragments, we know we've found live Lophelia when we find a bunch of rubble first. So we tend to look down in front of the ROV, and as soon as we see that rubble, we know to look up for the live colonies. So this is a zoom of some of that rubble. The white coral is actually, those are the fragments that are still alive. They may continue growing, they may get buried by other rubble and also perish. And then the next shot will be the similar pieces, but the polyps are out, so they're feeding structures. So then when we got to the top of this rocky outcrop, this is what we found. We saw a lot of shy rockfish, but we also saw a ton of live Lophelia pertusa. And so that was really wonderful to see and to note that there were a lot of fish around this type of coral and within this habitat. And so we were able to identify some of those fish species we likely saw on the sonar from previous surveys. These are some other highlight photos where we saw black corals, a lot of anemones, and in some areas 100% cover like those previous surveys. The next area I'm calling deep footprint reef. Since I was the PI for some of these dives, I got to come up with some of the names unimaginably. And so this is a rendering of the approximate track that we took. This top ridge in the map on the top right is the very well-studied ridge. So we wanted to extend those surveys down deeper to see if there are any differences in depth between some of those on that ridge. And so these are some highlight photos, and then I have this video. So we dropped down to 500 meters, and we immediately saw that we were in a high-density coral and sponge garden, which was very exciting. So there's a lot of hard bottom habitat here, plenty of spaces for deep-sea corals and sponges to attach and grow. There tended to be a lot on these big rocky outcrops. Like I mentioned before, they like to be up off the bottom so they can intercept as much high current as possible to increase the rate at which they can filter feed. Then we got to the top of the ridge, or almost to the top of the ridge, and we saw another coral garden of this Acanthogorgia or golden sea fan. And they were quite dense. There were a number of other species here too, including bivalves that we saw. You can see a few here. There are sponges. In the upper left, there's a basket star. So a really beautiful place, and one of the reasons why it's a really well-studied area. Unfortunately, we did have to abort this dive early. We saw a lot, and we encountered a lot of lost fishing gear. So this includes traps, lines with floats on them, and when you're driving an ROV around, it's very dangerous to have a tethered, very expensive piece of equipment be potentially tangled into some of these lines. And so we saw a lot there. And this is, even though this is within the Footprint Marine Reserve, so an MPA, these traps and pots and lines probably got brought in by current and snagged on all these rocky outcrops that you're seeing. So then I think the most exciting dive that we did was close towards the end. So we're back in this area again, the ridge being footprint, and then this big bank below it is known as Piggy Bank, the other really well-studied area. Overlaid on this map are all the previous surveys done by the National Marine Fisheries Service, as well as some of the sanctuary surveys. And you can see within these highlighted areas, the tops of these seafloor features are some of the best studied. There's a high density of all of these tracks that the submersibles have done. And then overlaid on that is the deep footprint transect that I had just showed you previous. And so this next dive was even deeper than that, in between the two really well-studied features. So one of the reasons why there's kind of a depth limit of those surveys, those previous surveys, is because those submersibles were limited to about 350 meters. And so they couldn't go any deeper than that. So with the Ocean Exploration Trust's ROVs, we could go down to 1,000 meters. And so we took advantage of that technology. And we discovered some really amazing and novel habitats that we've not seen this far south. So we went back and we landed in another coral and sponge garden similar to that first dive, since it's in a very similar area. There was a high density of these individuals. They were large individuals. They were quite diverse. But the most exciting part was this glass sponge graveyard that we came across. And so all of the porous material that you're seeing that's brown is dead glass sponge. Glass sponges are unique in that when they die and their living tissue sloughs off, their skeleton is made out of silica and it remains post-mortem. And it's a hard fused skeleton. So unlike calcium sponges that would disintegrate eventually, these leave behind a hard surface that other species can grow on top of, so they can attach and grow. Some of these fluffy cloud-like puffballs that you're seeing are live fringes of this glass sponge. It's called Pharea. And so we saw a bit of the live sponge, but it was mostly dead. And this is a close-up of some of that frilly cloud sponge of Phorea. And if you were to take a bunch of this in your hands, it would crunch and it would crumble like potato chips. So it's a hard skeleton. And there's a lot of organisms that can live and grow within this kind of habitat. Sponges, brittle stars, crabs, fish use it for protection. So we saw a number of other species utilizing this habitat. And this is the furthest south we've ever observed this type of potential reef. We're not 100% sure if it's an actual reef or not, but the most similar kind of habitat that we've seen so far like this was observed in British Columbia. So this is quite novel and really an awesome discovery within the Channel Islands Sanctuary, and I think it really just highlights why exploration and characterization is so critical. This lays between two of the best studied points within Southern California with respect to deep sea corals and sponges, and we had no idea it was there. And so these are just a few more pictures here of some of the highlights. And we're actually partnering with the National Marine Fisheries Service. They are out on our research vessel now, the Shearwater, and they have an autonomous underwater vehicle, and they are going back to try to map the extent of this landscape of dead glass sponges, because despite having an 8-hour dive here, we never reached the end of it. And we were meandering and zooming in and collecting samples, but it's quite extensive, and we're very excited to have some of that data come back so we can learn more and ask more questions. And so with that, I'll say thank you. We couldn't have done this without a number of partners. It certainly takes a village, especially with such expensive and harsh research that we do in harsh conditions. And so thank you so much, and I'll take any questions you might have.
Hey, Lizzie, that was awesome. That was wonderful. Oh my gosh, you were fast too. You speak very fast. So our first question is from Jason. And that is, did your team consult or assist on the Monterey Bay Aquarium's forthcoming Into the Deep exhibit? Is the emphasis on West Coast corals and sponges?
Yes, actually. They reached out to us, and we met a few times to talk about some of our work, and we collaborated. We invited the Monterey Bay Aquarium to come out with us on one of the cruises that was funded by the West Coast Initiative that occurred within the Channel Islands Sanctuary, and we were able to collect some live corals for them to do husbandry experiments. And so I do believe they're focusing on West Coast region corals, but I also think they might be getting specimen potentially from Alaska, which is where the next initiative is happening, the Alaska Deep Sea Coral Initiative. So yes, we've been in conversation, and we're hoping to work together on into the future after the initiative is over.
Wonderful. Will you be doing any work in Alaska? You are going to be involved with that then?
I won't be. We've been kind of passing on everything we've learned, and so we have coordination meetings with those leads and coordinators within Alaska, and so we work together, but I won't personally be doing any work there, unfortunately.
It's too cold up there anyway.
It is.
So next question is, how did you define deep sea? What depths or range?
That's a great question. Since I work for NOAA, NOAA has a definition of 50 meters or deeper, but of course deep sea corals and sponges don't sit right at the 50-meter mark if they're going to be considered deep sea. And so some of them, I think they have to live at least 50 meters or below to be considered by NOAA deep sea corals and sponges, but they can certainly extend their range into shallower depths. And that's particularly true in higher latitudes because the water is so cold at shallower depths. Some of those ranges actually extend. So in Alaska, deep sea coral and sponge species that you would find in several hundred meters of water here on off the coast of California actually extend up to only 50 or so or less meters because the water is just so cold there. So they can actually survive at shallower depths.
Wonderful. I think the water is cold here. Imagine up there. So our next question is from Andrew. Given the darkness, is there any evolutionary advantage to deep sea corals being so vibrant and colorful, and/or colorful?
I've been asked this question before, and I don't have a good answer. They come in a range of colors from red, orange, pink, white, yellow, purple, and because light doesn't reach that far, there's really no organisms that can see it, so they wouldn't want to necessarily hide or blend in with anything, but it might just be something, the structures within their tissues happen to be that kind of color, but I don't have a great answer for that, and I hope that one day I will.
All right, let us know when you do find out. Okay.
I'll come back.
All right, deal. Jessica would like to know: is the bed of glass coral you saw considered unhealthy compared to similar other beds around the world?
That's also an excellent question. So those glass sponges are indeed dead, and there could be a number of reasons why that potential reef died, from increases in temperature that happened a while ago that killed them off, maybe an increase in ocean acidity from an extreme upwelling event, maybe a landslide where a sediment plume came and kind of smothered the sponges. But we don't know why, and I wouldn't say that they were necessarily less healthy, because there were a number of other sponges growing on top of it, and right beside it was a coral and sponge garden that we found. And so one of the questions we're asking is, are the fish and invertebrates that associate with those dead glass sponges the same or different from the adjacent coral and sponge gardens that we saw? And so I think at some point it was thriving. I wouldn't say it's necessarily unhealthy now because the conditions have changed and evolved, but there also are dead glass sponge reefs up in British Columbia. They have both living and dead, but so far that's the only place in the world, I believe, that they've been observed. And so this could be a really interesting plot twist in terms of the conditions that they need to grow.
All right, thank you. So it's almost like, I know in our wine country they have these little microclimates, so there could be those little microclimates underwater as well, huh?
Definitely.
All right, so Diana would like to know, when you came across the fishing gear, is it possible to remove them without damaging the reef?
Another excellent question. It would be really difficult to remove them, and I do believe there is a group that whose mission is to remove deep sea debris to an extent. Deep sea work is very expensive, and so that kind of removal would be very technically difficult because a lot of the piloted technology that's used is tethered to the vessel, and so just the risk of entanglement would be great and potentially not worth the risk. It would be expensive. I think it would be possible, but depending on the type of entanglement that's happening with the reef, it would probably damage the reef as well. So it's an unfortunate situation, but I think in some cases it's possible, but it'd be difficult to find all of that gear, and it would be difficult to devise a methodology that would safely remove it for the reef as well as for the technology you're using to dislodge it, I guess.
All right, thank you. Laura would like to know, how healthy are these coral and deep sea sponge communities as compared to the more surface coral reefs? Have these shown an effect of global warming?
They have. And so bleaching can also occur in deep sea corals, just as they might in shallow water corals and sponges. But it's more because they're losing their tissue. So some of them, when they turn white, it's because it's just their exposed skeleton. And it's hard to say. The deep sea is a pretty stable environment, and a lot of the species are adapted to a very narrow range of conditions: temperature, pH, and so even the smallest perturbations to those conditions could cause mortality or stress events in those kinds of species. There's a lot of widespread bleaching happening right now in shallow water corals and sponges because the surface is just so more variable than the deep. So I think, and especially with the storms, I think that might actually put shallow water corals in the more damaged or threatened habitat just because they're exposed to those storms that are happening at the surface. But deep sea corals and sponges are not far behind. With increasing temperatures and the heat wave, if you recall the blob that happened in 2016, 2017, we have a researcher that had put temperature data loggers out in the Channel Islands Sanctuary down to about 200 meters, and they were able to record anomalously warm temperatures down to 200 meters. And so they're not completely out of touch with things going on at the surface either. So.
Was that blob that when there was like that milky white band of water?
So the blob was actually like a marine heat wave, and that happened all throughout the eastern Pacific, from the center of the Pacific all the way to the west coast.
Because I remember about that time, 5-6 years ago, there was this milky white kind of band of water, and one of our commercial fishermen, Mike McCorkles, and he's been fishing here for 60 years, I think, had never seen anything like that, and it was warmer water. It was really a strange thing. Okay, so another question, and unless anybody else has questions, please type them in. This could be our last one. From Don, again, our passport president, who used to sign my paycheck, so I always ask his questions. What impacts are researchers seeing with deep sea corals and shallow corals as a result of the gradual CO2 rise in oceans?
So an excellent example of that, let's see if I can go back. Yes, okay. So the Lophelia pertusa species that I had mentioned, with the rising CO2 in the atmosphere being absorbed by the ocean, increases the acidity of the water, and that actually can deteriorate the calcium carbonate skeletons for stony corals, which is what you're seeing in this video and then the bottom left photo. And for this particular, normally a reef building species, it's just making it so weak that it grows polyps and then they crumble and fall off. And so it creates a lot of rubble at the bottom versus being a reef building coral. And so for the stony corals, that's been a big problem, and there aren't that many on the west coast in the deep sea that are really habitat forming. It's more of these soft sea fans in the upper left and in the middle bottom photos. So those are becoming the more dominant species. So there could be ecosystem service shifts depending on the communities, from what they used to be to what they're turning into now. But a lot of stress, their immune systems may be compromised, so they're more likely to be taken over by zoanthids, which are a parasitic kind of anemone that can grow on the skeletons of some of these sea fans. So there's impacts, and we're still trying to figure out exactly how they'll respond because there's so little known about their biology and physiology. It's hard to do husbandry experiments, it's hard to get them, it's hard to replicate those conditions at the surface. And so we know there's going to be impacts, but we just don't know quite what they are yet, other than this pretty famous example of this Lophelia coral.
Thank you. So we do have one more question. Ruth wants to know, normally in surface corals, the corals are a cooperative organism of algae and the corals. Is there a cooperating organism in the deep sea corals?
So they don't have those symbiotic algae because they live at depths where there's little to no light whatsoever. But there is research that's beginning that's looking at microbes that live on these corals and sponges that might serve some kind of symbiotic purpose as well, where the coral is benefiting from having these microbes, potentially their immune system. So depending on the community of microbes that live on their surface or within their tissues, they might be better at fighting off disease. But again, that's such a new area of research. We don't know a whole lot about it yet. So no algae, but potentially microbes and maybe even other things we have yet to discover.
All right. Well, thank you. Thank you so much, Lizzie. We greatly appreciate your talk tonight. Very informative. We do have a little gift bag for you. I'm going to bring that out to you in just a second. And we hope to see you back here next month for the talk on the desalination plant. Again, Lizzie, thank you very much, and everybody have a great evening.